Welcome back to Falcon Physician Reviews. In Module 2, we'll be discussing fertilization through implantation. Let's get started. You're paged to see a 22-year-old sexually active woman in the ER who reports missing her regular cycle for the last 8 weeks. She's complaining of acute onset of lower left quadrant pain in the abdomen, and being an astute and recent graduate, you assume she is experiencing what until proven otherwise? Think about your answer, and we'll come back with this in a second. The answer we're looking for is D, ectopic pregnancy. For your boards, they're going to be interested that you know the most common sites of ectopic implantation. For that, we'll look a little bit closer at the structure of the fallopian tube as it relates to the uterus, the typical site of implantation. If you have the fimbriated end of the infundibulum, this typically would be over top of the Graafian follicle, so at the release of the oocyte, then it will be taken up into the fallopian tube and moved down through that tube so that it can implant within the next couple days into the uterus itself. However, if there's some kind of blockage or something preventing the movement of that oocyte as it gets fertilized by the sperm, it's going to have difficulty and get trapped. Typically, it will get trapped and implant at the ampulla, and this is where it will implant for the tubal pregnancy. Less commonly, it can get trapped outside into the peritoneum, and there can be a peritoneal pregnancy, which can be equally as dangerous because of the vascular flows that will set up later in pregnancy. However, it has been known that there's been a full-term pregnancy carried in the peritoneum of a female and then birth through cesarean section. Nonetheless, your questions will probably focus around the site of most common fertilization for tubals, which will be the ampulla. Now, looking at the endometrium, we want to discuss the layers of the endometrium, which will be the compact, spongy, and the basal layers. The compact and the spongy layers will form the functional layer, and this is developed in the final stages of the menstrual cycle. The spiral arteries will continue to grow through, glandular growth will occur from the uterine gland, and this functional layer is in preparation for implantation into the uterus, which typically occurs around the fundic region. If there is no implantation because fertilization did not occur, then it's the layer between the basal and functional layers where the spiral arteries will retract, hydrodissect away that entire functional layer. So, looking at fertilization, we know that fertilization is a fusion of the male and female gamete to form a single cell called the zygote. We've been reminded that the normal site of fertilization is the ampulla, but it's also a site of common tubal pregnancies in clinical scenarios. And we know that there's multiple opportunities for fertilization and that they require a large number of sperm from the male, typically 200 to 600 million, with only 200 or 300 reaching the oocyte. And you can see by this drastic drop in those that actually make it to the ampulla for fertilization, with only 50 million sperm, which can only be retained for about 48 hours with being functional. It makes it very unlikely, but not impossible, for someone to get pregnant. So they become essentially infertile and not actually infertile. Looking at the first week of fertilization, we'll move the zygote through and take it down its first stages. So assuming that a sperm does make it to the oocyte, the first stage is that there's going to be a zonal reaction to block polyspermy as soon as the first sperm penetrates into the zona pellucida. Then the male and female genomes will become and make the male pronucleus and the female pronucleus. Notice that the tail of the sperm and all of the mitochondria associated with it will degenerate, and this is why maternal mitochondrial DNA is what is passed on from generation to generation. It's that first point right before the pronucleus of the male is produced that the female will finish meiosis and discard that second polar body. In fertility clinics, they'll use this fact to tell whether or not the sperm has actually been successful at making the zygote. In the third stage, the fusion of the pronuclei will then be responsible for the formation of the zygote. And then finally, they'll start to replicate their DNA by fusing together, and it's the female genotype that's activated first for these first cell divisions that will occur. So as we look through the different stages of division, we're going to take it from the 1-cell stage through to the 8-cell stage. And what's going to happen here is that the zygote will be inside the zona pellucida and not sticky, which is the timeline that it should take to travel down the fallopian tube for the first 5 or 6 days. If it can't travel down the fallopian tube, this is the point, once it hatches, that it will implant into the fallopian tube and have a tubal pregnancy. During this stage, it's important for you to remember that the cell size has been set already. There's not enough time for it to reproduce and make more cellular fluids. Therefore, this huge oocyte is going to be bringing in all of the cytoplasm that these first few divisions will have to use. But this is important because it restores the actual normal size of the cells by dividing through this first set of divisions, which will take it all the way to the blastocyst stage. The next stage, from 12 to 16 cell stage, is called the morula, which resembles the raspberry or mulberry. And after the morular phase, it's going to start to form an inner cell mass, which will give rise to the embryo proper, and a peripheral layer of cells called the trophoblasts, which are the first cells to make a division of becoming potentiated. So the trophoblasts are indeed the first set of cells that become differentiated into a specific type of cell. Up to this point, especially through the 1 to 8 cell stage, they are considered pluripotent and omnipotent. As the morula starts to divide more, the blastocyst will be formed, and this is identified by the development of a fluid cavity that will appear around day 5. This is going to be fluid that's seeping inside the morula, and the space is going to become compacted and form the blastocelic cavity. And the inner cell mass will then start to contact one pole of the trophoblasts. That will become the embryonic pole. And it's that embryonic pole that they'll be talking about for the implantation phases in some of the questions that you could be asked during your step 1 preparation. Now, this blastocyst is going to be around day 5 after fertilization, and it's now getting ready for implantation. So the zona pellucida has to disappear, and it starts to break down. Once it breaks through or hatches from the zona pellucida, it's ready for implantation because it's become sticky. And it's at this stage it has about 107 to 110 cells, and 99 of those will become placenta, and only 8 of those will become the embryo itself. I think this is a point where you should star this, because oftentimes this will be a question that can be asked of you as far as how much of this early blastocyst will be responsible for the formation of the embryo. So let's look at this graphically. We have the 2-cell stage where the maternal genome has been activated. They're very large cells at this point, and they're still within the zona pellucida. As it starts to go into the 4-cell stage, remember these cells have omnipotence and can make any tissue they want, from trophoblast to embryo. Then we get into the morula, the blastocyst, which will now have the 8 to 16 cell stage, still within the zona pellucida. And as that morula starts to compact towards the end of the third day, it will start to add fluid to it, where they'll get the early blastocyst and the trophoblast starting to form around the perimeter with an inner cell mass at one pole. The zona pellucida is starting to degenerate at this point, and they think that might be one of the pathways through which it can draw in fluids from the surrounding tissues. And then finally, in the late blastocyst stage, the zona pellucida will disappear and implantation is now possible. As it makes its way down towards the fundic region of the uterus, where the typical implantation site is, the endometrium should have already been prepared. If not, and there's no site for implantation, then the early embryo will then just pass through and the menstrual cycle will begin. And it's believed that many pregnancies never make it to the implantation stage because of hormonal or genetic problems, which make it impossible for it to survive. It's estimated that up to 50% of those are actually spontaneously aborted. Here we can see that the embryonic pole has implanted into the endometrium, and it started to proliferate the trophoblast into two cell populations. There's the syncytial trophoblast, which actually has no cellular borders and is rather amorphous blob of very invasive type cells. And there's the cytotrophoblast, which will be the inner layer, and that will end up separating the villi that will form along the early placenta. This early embryo will actually be entirely implanted over and into the endometrium in the early phases. And it always implants with that embryonic pole inwards. The syncytial trophoblasts are going to be responsible for eroding the endometrium via enzymes, and they'll produce the human chorionic gonadotropin that will be an early indicator in some of the pregnancy tests that are available. It will actually burrow into the endometrium and start invading it, and then there's an important decidual reaction that occurs with the very glandular cells that have been prepared by the maternal hormones, and the glandular tissues that are evading and being eaten away by the syncytial trophoblast will actually serve as an energy source for the developing embryo because of the proliferative growth rate. Once it gets a little bit further into develop and some amniotic cavity fluids now start to be drawn in from the embryonic pole, there will be development into two distinct cell layers of the bilaminar embryo. The epiblast, which will actually form all of the germ layers, and the hypoblast, which will be responsible for forming some of the yolk sac and some of the membranes, such as the exocelomic membrane that will line the cytotrophoblasts. As we move to implantation in the bilaminar embryo, we've talked about the fact that the blastocyst will hatch from the zona pellucida, which makes it sticky. There'll be then chances for ectopic pregnancy if it has not made its way to the uterus, or if it implants somewhere other than the fundic region, for instance over top of the cervix. It's known as a placenta previa, which is a high-risk pregnancy, since the placenta needs to be broken and passed through the cervix during pregnancy, or it can be susceptible to early injury because of being open to that cervical os. The trophoblasts are going to be the first cell population to differentiate, and they'll make two populations of cytotrophoblasts and syncytial trophoblasts, and it's the syncytial trophoblasts that are invasive and less defined cells. The blastocyst will implant for nutritional survival, so if it cannot implant and go through that early invasiveness of the syncytial trophoblast, it will be spontaneously aborted. And one important feature of this is that it goes through the decidual reaction, which is an endometrial stroma that becomes high in glycogen and other enzymes, which will counterbalance the invasiveness of trophoblast. So if the decidual reaction does not occur properly, there's the potential for the trophoblast to become hyperinvasive and actually cause some choriocarcinomas. Fluid, which will be the embryotroph, will supply the nutrition to the conceptus during this early decidual reaction. And the maternal leukocytes, which will be secreting interleukin-2, is an important feature for you to remember for testing, because it's this interleukin-2 that will prevent the rejection of the early embryo and produce an area of immunologic sequestering for the embryo's development. Once you have the bilaminar disc, you have the inner cell mass, which will organize into two cell layers, the epiblast and the hypoblast. And they'll start to take in fluid, which will be the extracoelomic cavity or chorionic cavity, as well as the amniotic fluid, which will be at the polar end of the embryo. The extraembryonic mesoderm will migrate out and form the primary and secondary yolk sacs, and those will be derived from the epiblast layers. We've already discussed some of the possibilities of spontaneous abortion and how many of those are actually ill-fated from the beginning, and it's believed that this is nature's process of aborting embryos which are ill-fated from the beginning. This concludes section 2 on fertilization through implantation. The next section will deal more detail on the placenta formation and the fetal membranes associated.